In this session, we start with the structural representation for the protein. Atoms actually not easy to make a picture. As you know, it could be particle or and wave, and also it could be explained by the quantum mechanics and also some the statistics. In chemistry, most popular models are space filling model and ball and stick model. In this picture, you see ball and stick model in A for the myoglobin. Main chain plus side chains only on the surface. However, by looking at this model, it's very confusing, and you can get little information from this structure. So we need another way to make structural pictures. The rest of things are made by computer by authorless. B is oversimplified. You just trace main chain roughly with him. You can see clearly where him the cofactor is positioned in overall structure. In C and D, the main chains are more elaborated. You can trace. Even in D, you can see clearly the alpha helix by the column shape. Jane Richardson proposed another way, the helical ribbon for the alpha helix and the arrow ribbon for the beta pretty sheet, and the loops connecting such alpha secondary structures. Sometimes it's very useful to draw emphasizing the secondary element, especially for beta. One of such drawing is called topology diagram. By here you can see the number and relative direction and connectivity of secondary structure, mostly for the beta pretty sheet. This is very useful to compare different protein structures. You see above the real protein drawn by Jane Richardson model, at the bottom topology diagram. So you can see here 4 betas are connected by anti-parallel, and also you can trace the relative connectivity between them from N to C, things like that. B and C are much complex, but topology you can see more clearly. Another method is called TOPS diagram, using triangles and circles. Triangles two ways, regular or upside down. So regular indicates strength just coming from face towards you, and upside down triangle in opposite direction, from you to page. Circles represent helices, the real protein, and we can trace by this TOPS diagram. N terminal and alpha helix, and then connected to beta, which comes from page towards U, and connected to another one that comes from U to page, something like that. This one is not explained in our textbook. Next, label will be motif or super-secondary structure. More than two secondary structures they get together with specific geometric arrangement and appears in many proteins over and over. Sometimes it provides particular functions, some they don't have. First example is helix turn helix. Two helices are connected by loop. The first example was found in the albumin, which is muscle protein. This protein has three alpha loop, three helix loop helix, one of which is called EF end. You see, two helices are connected by loop, where calcium ion binds to. You can see the actual configuration formed by three fingers, and the calcium position in a pocket made by three fingers. In B, you can see calcium binding bonds between the protein and calcium, negatively charged aspartic acid D and glutamic acid E, side chains is key element. In addition, there are some main chains. The peptide bonds also contribute in binding. In C, this is troponin C, present in muscle, binds to calcium reversibly, as you see here. In this slide, you can see the sequence constraint to make EF end. Three different proteins having EF end aligned. As you see at the bottom, two helices where many hydrophobic residues are highly conserved in the green color. Actually, two helices interact each other, making hydrophobic bonds. Loop also has the highly conserved negatively charged D and E in yellow color, providing the calcium binding site, as I mentioned. In middle, you can see glycine, which also highly conserved, allowing the sharp turn. Next motif will be hairpin or beta-beta unit. Two antiparallel betas are connected by a small loop. In B, you can see two hairpin structures in single protein. Next one will be another complex but beautiful motif called Greek key motif. So basically, four anti-parallel betas are connected in this way. 1, 2, 3, 4, making certain drawing found in Greece antique. You see, the real protein called staphylococcus nuclease, how it can be formed. Long stretch of hairpin with loosely formed at the middle, allowing fold, making this kind of structure. 4 betas could arrange it in so many different ways, actually 24 different ways. Only 8 shown in 2 layers top appearing in real proteins. First 2 belongs to Greek key. Okay, for some reason only limited arrangement connectivities are only appearing in proteins. Next one will be beta alpha beta motif. 2 betas are connected by alpha and also 2 loops. First loops are providing functional site. Second one doesn't have. You can see another view. There are 2 ways the alpha helix is present above the plane made by 2 parallel betas or below. Only above structures are present in real protein. This is called right-handed. Next level will be domain. Domain could be defined as independent folding unit. This indicates domains could present alone. Okay, EF has a single domain, but urokinase has 3 domains. Factor 9 has 4 domains, something like that. So all of these proteins only have a single polypeptide. So only one tertiary structure, however, composed of single or multiple number of domains. So each domain could exist alone. Even though you cut here, they could exist. In this picture, you can see the protein structure of TIM, tyrose phosphate isomerase, that has 4 repeating motif, beta, alpha, beta. So with different colors, yellow, red, things like that. In real 3-dimensional structures, they present in a row, yellow, red, green, things like that. Okay, that happens very often. Proteins could be classified 4 different types based on the secondary structure worked by Levitt and Jostia in MRC. First, alpha protein. This is hemoglobin, having only alpha helices as secondary structure. Beta protein, only beta pretty sheet. Alpha plus beta and alpha slash beta, they do have both. Difference between 2 of them is alpha regions are separated from beta regions. That's going to be alpha plus beta. 2 structures get together, mixed. That's going to be alpha slash beta. Lastly, we could summarize hierarchy of protein structures. Motif and domains are additional in traditional structures.